Welcome to Lesson 11. In Lesson 10, you learned about dependent samples or repeated measures. Just to refresh your memory, that could be where we give the same person two different conditions to see how they react to each one. Maybe a control and then a treatment, or maybe two types of treatments. Or this could be longitudinal, where we measure some variable at one point in time and then again at another and see if the variable changes. Or this could be a pre-test and then post-test. What was the measurement of a variable before and after a treatment? These are just a few situations in which we would use dependent samples. This type of research design is really useful because it controls for individual differences. In other words, if we gave someone some kind of treatment, then those same individual differences will be present the next time we give that same treatment. That way we can see how two different treatments play out under the same conditions. Because this controls for individual differences, we can then use fewer subjects. And this is more cost-effective, less time-consuming, and generally less expensive. However, there are also a few disadvantages, one of which is carryover effects. For example, let's say we have this new method of teaching math. You want to know if it's going to be effective. If you use the same group of students to test this new teaching method, inevitably they're going to be better at math the second time around. So if the first time we teach them one way, and then the second lesson we teach them a different way, they'll already be better at math from learning it the first time. Then we don't know if the results after the second treatment are due to the fact that it was effective, or due to the fact that they've learned math before. That's just one example. So the second measurement can get affected by the first treatment. And the order in which we give the treatment might influence the results. For example, say we want to test two types of pills. What if the first pill has some kind of interaction with the second pill? And so by taking it in that order, they affect the results. Therefore, in this lesson, you're going to learn about independent samples. Whereas dependent samples deals with within subject designs, independent samples deals with between subject designs. In this case, the advantages of dependent samples are the disadvantages of independent samples, and the disadvantages of dependent samples are the advantages of independent samples. Does that make sense? With independent samples, we need more subjects, because we need to randomize the two groups taking the two treatments. We need a larger N to control for individual differences, as best as possible. That means it's more time-consuming and generally more expensive. But then the advantages of independent samples are that we don't have carryover effects. Therefore, we can give one treatment to one group, another treatment to another group, and not worry about one treatment affecting the other, because each person or each subject only gets one treatment. With independent samples, we can do an experimental test where we give treatments to the subjects, or observational, where we simply observe characteristics of two different populations and then compare them. Everything is exactly the same. The null and alternative hypotheses, the t-statistic, and the way we make our statistical decision.